Welcome back! Now that we've explored transcription, it's time to complete the journey by diving deeper into RNA splicing, practice problems, and of course, the process of translation, where proteins are born. Let's pick up where we left off. After transcription, the newly formed pre-mRNA isn't quite ready for action. It contains introns, long sequences of nucleotides that don't code for proteins. Think of introns as the deleted scenes of a movie, unnecessary for the final cut. These are removed in a process called RNA splicing. What remains are the exons, the shorter sequences of nucleotides that carry the real instructions for building proteins. Once the introns are spliced out, we're left with a finalized mRNA strand ready to head out of the nucleus and into the cytosol. Let's try a quick practice problem to understand how DNA and RNA are linked. Imagine we're given a DNA strand running in the 3' to 5' direction. How do we transcribe the corresponding mRNA strand? Here's a quick guide. G pairs with CC pairs with GA pairs with U in RNA, since RNA uses uracil instead of thymine. And T in DNA pairs with A in RNA. Let's try. If the DNA sequence is TACGTA, the mRNA sequence will be AUGCAU, written in the 5' to 3' direction. That's how the genetic code gets transcribed. Now, let's talk about translation, the process where mRNA's instructions are decoded to construct proteins. Once the mRNA exits the nucleus, it enters the cytosol and meets up with a ribosome, the cell's protein-making factory. Ribosomes can either float freely in the cytoplasm or attach to the rough endoplasmic reticulum. Translation begins when the ribosome clamps onto the mRNA strand. Let's break this process down into its 3 key steps: initiation, elongation, and termination. Step 1. Initiation. The translation process kicks off at the start codon, AUG, which signals the ribosome to begin. This codon matches with a specific transfer RNA molecule carrying the amino acid methionine. The tRNA's anticodon, UAC, pairs with the AUG codon in the ribosome's P site, also known as the peptidyl site, where the first peptide bonds are formed. Step 2. Elongation. Next, another tRNA molecule enters the A site, the aminoacyl site of the ribosome, bringing the next amino acid. A covalent bond forms between the amino acids, linking them into a growing polypeptide chain. As the ribosome moves along the mRNA strand, the tRNA in the P site shifts to the E site, exit site, where it releases its amino acid and exits the ribosome. Meanwhile, the tRNA in the A site shifts to the P site, making space for the next tRNA in line. This cycle repeats, lengthening the protein 1 amino acid at a time. Step 3. Termination translation ends when the ribosome encounters a stop codon, UAA, UAG, or UGA, on the mRNA strand. These codons don't code for any amino acids. Instead, they signal a release factor to enter the ribosome's A site. This release factor causes the ribosome's subunits to disassemble, releasing the newly formed protein. The protein then heads to the Golgi apparatus for folding and modifications, shaping it into its functional form. So, to recap, transcription converts DNA into a finalized mRNA strand, ready to leave the nucleus. Translation uses that mRNA to build proteins with the help of ribosomes and tRNA molecules. Thanks for joining us on this journey through transcription and translation. Be sure to like, subscribe, and hit the bell for more deep dives into biology and beyond. Until next time.